Hello everyone. We are going to discuss the new topic, and the name of topic is reproduction in plants. Before that, let us discuss what is reproduction. Reproduction is the functional ability of all living organisms by which they used to produce their own kind. This is important to continue their race. Now, reproduction is basically divided into two types. First is called as asexual reproduction, and the second one is called as sexual reproduction. Asexual reproduction is mostly seen in lower organisms, and it is characterized by uniparental procedure or uniparental mechanism in which single individual is responsible for formation of new individual. Now this process includes only mitosis. No meiosis is involved, and therefore gamete formation is not seen in asexual reproduction. Gametes are not formed, so there is no chance of, or there is no occurrence of fertilization, and therefore new individual which is formed from a single parent by means of or without formation and fusion of gametes, and therefore the process is called as apomixis. Apo means without, mixis means mixing or fusion, or you can call it as fertilization. So the newly formed individual, they are exactly similar or genetically identical to that of the parental individual, and therefore they are called as clones. So the result or outcome of asexual reproduction, it is called as clones. On the other hand, sexual reproduction is a biparental process, and in this process both the parents, that is male as well as female, they require. Now these parents undergo meiosis, or they show meiosis for gamete formation, and these gamete forms or these gametes formed during the process, they show fusion or mixing, and therefore sexual reproduction is also called as amphimixis. Amphi means both male as well as female gametes, they fuse together to form the new individual, and due to meiosis and combination of the gamete, fertilization of the gamete, the newly formed individual, they show variations, and therefore the outcome of sexual reproduction is variation, and hence sexual reproduction is evolutionary important. This part is very essential to understand, because variation is very essential for evolution, and therefore sexual reproduction is having the role in evolution, and asexual reproduction is having no role in evolution. So guys, let us discuss different types of asexual reproduction. Asexual reproduction in lower organism is mainly divided into following types. The first one is called as binary fission. Binary fission is mostly seen in unicellular organisms. Unicellular organisms we can commonly say in kingdom Monera as well as kingdom Protista. So you can say bacteria and some protists, they used to show binary fission. Here single cell or unicellular parental organism, it will show the cell division obviously by mitosis. It will show a constriction like this, and this parental cell due to nuclear division finally divided into two new individuals. This process is called as binary fission. The best example of binary fission is bacteria and protist like amoeba. So this is the first type of asexual reproduction. Now the second type of asexual reproduction is budding. Budding is nothing but a bud formation. This is the external protuberance developed from the body of parental organism. See, for example, this is the parental organism's body, and from which the external bud is formed like this. Now this parental nucleus will divide to produce a daughter nucleus, and this protuberance will grow gradually and finally get detached from the parental individual to produce new individual like this. So this is the result. Two individuals are formed from one parental individual, and therefore this is also called as asexual reproduction. Budding is mostly seen in the organisms like yeast. So this is the second type. The third type is formation of spores. Different organisms, different individuals, you can say like alga, fungi, bryophyte, pteridophytes, they used to produce spores. Spores are of two different types, asexual spores as well as sexual spores. Now here we are going to discuss about only asexual spores produced by certain organisms. Now spore formation or asexual spore formation is mostly occur by means of motile as well as non-motile spores. Now there are two types of spores that is discussed. First is called as zoospores and the second is called as conidia, or you can also call it as conidiospores. Can you guess which out of these two is motile type of spore? The answer is very simple. The one which is indicated by zoo will show the motility, and therefore zoospores are the motile spores, and these spores are mostly produced by the organisms like algae. Now these spores are having flagella or cilia, and those are essential for lashing movement, for swimming of the spore, and therefore zoospore is characteristically seen in algae as they are showing aquatic habitat. The second type of spore, which are the asexual type of spores, are called as conidia, and these conidia are non-motile. They will not show locomotion. They are not supported with flagella and cilia. They develop from the filament and produced in the chain like this, which is dispersed by means of wind, by means of water to different places and involved in the formation of new individual. Formation of conidia is the characteristic feature of fungi. Different kind of fungi, they used to produce these, or you can say conidia, as the mean of asexual reproduction. So this is the third type, spore formation. The last type is called as fragmentation. The fragmentation is nothing but formation of small individual fragment from the main parental body, main parental organism. Fragmentation is the type of asexual reproduction which is mostly seen in the organisms which are having filamentous bodies. Now let us discuss suppose this is the filamentous body of the individual, which is mechanically broken down due to certain reasons. Many reasons are there. Due to certain reasons, this body is broken into fragments. Now this individual fragment follow the polarity. When this fragment is dragged away and will found the substratum like this. Now suppose this is the top region and this is the lower region. When this lower region will come in contact with the soil or the substratum, it will start developing the new individual like this. Now this fragmentation is responsible for formation of new individual asexually. And this kind of mode of asexual reproduction is mostly seen, as I have already told you, that fragmentation is seen in organisms having filamentous body. So the best example of fragmentation, they are algae, certain fungi and bryophytes. Now these four types of asexual reproductions are mostly common or commonly observed in lower organisms. You can see here bacteria, some protists, then fungi, algae, bryophytes. Those lower organisms, they used to show the asexual reproduction. But in case of higher organism, as far as we are talking about plants, there is also plants like you can say flowering plants, phanerogams, gymnosperms and angiosperm. Asexual reproduction is evident there also. But that kind of asexual reproduction is quite different, and we are going to discuss it in detail. Come on guys, let us discuss asexual reproduction in flowering plants, which is also called as vegetative propagation. Now, before discussing this concept, let us discuss the plant body and systems involved in the plant body. Actually, in last year's syllabus, there is one topic, morphology of flowering plants, and in that topic, you have studied the different parts of the plants. For example, the flowering plant or the body of flowering plant is divided into two distinct systems. One is called as positively geotrophic and the underground system, which is called as root system. The root system includes roots, and they includes the primary, secondary, tertiary branches with root hair. The second system is called as shoot system, and this shoot system consists of stem, which is the aerial part or the elongated aerial axis of the shoot system called as stem, which bears leaves, flowers, fruits, etc. So this is the body of the plant. Now, this body or the different organs of the plant can also be classified on the basis of their function. For example, root, stem and leaves. These three parts they are collectively called as vegetative parts, because these parts are not directly involved in the process of sexual reproduction. On the other hand, the organs like flower and fruit, they are involved in sexual reproduction, or fruit, this is the outcome of the sexual reproduction, and therefore these are called as reproductive part, or you can simply call it as floral parts. Now, see the concept carefully. Asexual reproduction in the flowering plant is also called as vegetative propagation. Propagation is nothing but multiplication, producing large number of individuals, and this propagation occur by means of vegetative parts only. Now this part is important. Now which are the vegetative parts? Vegetative parts of the flowering plant are root, stem and leaves, and when new individual is produced from either root, stem or leaf of the flowering plant, then it is called as asexual reproduction, because reproductive parts are not involved. Formation of gamete is not there, fertilization of gamete is not there, and genetically identical individuals are formed, clones are formed. Therefore, it is an asexual reproduction produced, or you can say new individuals are formed by vegetative parts of the plant like root, stem and leaves, and therefore asexual reproduction in flowering plant is also called as vegetative propagation. Now let us discuss what are the different types of vegetative propagation in flowering plants. Now vegetative propagation is mainly divided into two types. First is called as natural methods and the second is called as artificial method. Now these type of vegetative propagation you can observe in nature, and artificial methods are implanted, or you can say followed by man in agriculture as well as horticulture. We are going to discuss this part later on, but let us focus first on the natural methods of vegetative propagation. Now, as we have discussed, vegetative propagation, that is nothing but production of new individual, formation of new individual from vegetative part of the plant. And there are three vegetative part of the plant. One is root, second is stem, third is leaves. Now, on that basis, natural methods of vegetative propagation is also divided into three types, and those are vegetative propagation by means of roots, then by means of stem, and the last type by means of leaves. So, on the basis of number of vegetative parts in the flowering plant, obviously there are three: root, stem and leaves. Natural method of vegetative propagation is also classified into three types: vegetative propagation by means of roots, stem and leaves. So by means of roots, when roots are involved in the process of vegetative propagation, they have to fulfill certain criteria for that. Let us discuss. The roots which are involved in vegetative propagation are thick and fleshy. And this thick and fleshy nature of the roots involved in vegetative propagation is only or mainly because of storage of food material. So, roots which are involved in vegetative propagation, they show storage of ample amount of reserve food material in them. So, this is the first criterion. The second important thing is they must show the growing points. Now, what are these growing points? These growing points are nothing but the buds which are present on the roots. Now, importantly, you are already aware of that. Presence of buds is the characteristic feature of stem and not the characteristic feature of root. So, which kind of buds they are? You can give the answer because you have already studied this concept in morphology of flowering plant. The buds which are not present on the stem and present on other part than that of stem are called as adventitious buds. This is important. Which kind of buds are present on the roots? Then your answer is adventitious buds. Now see, roots when they are involved in the process of vegetative propagation, they are thick and fleshy. They show stored reserve food in ample amounts. Second, they show growing points in the form of adventitious buds. And so these roots are able to propagate the new individual, form the new individual. The well-known examples are, you can give the example, or I will advise you to go through the notes while you are observing the lecture. This content is very vast, exhaustive data is there. Whenever you feel that this concept is important, this line is important in your notes, I will advise you to underline or highlight that sentence. Here examples are very important, they can be asked for MCQ. So thick and fleshy nature of the root, then growing points in the form of adventitious buds are present, and such roots are involved in vegetative propagation naturally. The best example is sweet potato. Now this is the simple tuberous root. You can also take the example of fasciculated tuberous roots, for example asparagus, dahlia, etc. So these are the different roots which are involved in the process of vegetative propagation naturally. Now there is an exception, and what is that? See, roots which are involved in the vegetative propagation by natural method are thick and fleshy, and they are with growing points. Presence of growing point is very important. It is must criterion for vegetative propagation. But as an exception, certain non-fleshy roots, they are also involved in the process of vegetative propagation. This is also very important. As this part is unique, it carries higher weightage for your NEET exam and board exam. See, non-fleshy roots, they are also involved in the process of vegetative propagation. Now, must thing is, for vegetative propagation, root should have stored food material for the growth, for the development of new plants. This reserve food is very important. Then how can non-fleshy roots produce the new individual? The answer is very simple. See, this is the mother plant and these are the roots. Now this non-fleshy root, they bears the adventitious buds, and these roots are still connected with the mother plant. So provision of food as well as water is available from the mother plant, and so there is no need of excess stored food material in those roots. Now these bud will sprout during favorable condition, and they used to produce the new plant. When the connection is broken, it is cut down from the mother plant. Now this plant is able to grow as individual plant, and which is nothing but vegetative propagation or asexual mode of reproduction. As simple as that. So certain non-fleshy roots are also involved in the process of vegetative propagation by natural method. Best examples are guava, you are very well known of that. Then Moraiya in Marathi it is Kadi Patta, Albizia, and many more. So to state few, I have given some names: guava, Moraiya, and Albizia. These are the non-fleshy roots involved in the process of vegetative propagation by natural methods. The next and the last type is vegetative propagation by natural methods by means of leaves. Now when leaves, they are involved in the process of vegetative propagation, such leaves are succulent. Now succulent, the meaning itself indicates that these leaves are thick and fleshy. They show presence of storage food material, and water storage is also present in such leaves. Not only that, the second criterion, which is same for root and stem, they also show presence of the growing points, or they show presence of buds on leaves. So the leaves which are involved in the process of vegetative propagation, they are thick and fleshy, or you can call it as succulent there, and they show presence of buds. Which kind of buds are present on the leaves? Can you guess? Your guess is very correct. And the buds which are present on the leaves, they are adventitious kind of buds. As they are not present on the stem, they are present on other part than that of stem. So the buds which are present on the leaves involved in the process of vegetative propagation, they are called as adventitious buds. Now these buds, here we are going to use one more term for these buds, and this term is very important. You have to underline there, or you have to highlight, mark it out. It is very essential. The buds which are present on the leaves, adventitious buds present on the leaves, are called as epiphyllous buds. Here epi means above, phyla means leaves. The buds which are present on the leaves, they are called as epiphylous buds, or you can simply use the word foliar buds. The buds which are present on the leaves, the adventitious buds present on the leaves are called as epiphylous buds or foliar buds. Now these buds, they sprout during favorable condition. Let us discuss the best example of leaves involved in vegetative propagation, and that is bryophyllum. This is a very common plant. The leaf is like this with wavy margin, and in the notch of the margin there is epiphylous bud. So when epiphylous buds, here in case of bryophyllum, it is present on the margin. During favorable condition, these buds sprout to produce the adventitious roots and the plantlet like this. The best diagram is given in your notes. You are observing the notes there in front of you, and likewise many individuals have formed the single leaf, and therefore it is vegetative propagation by means of leaf. The example is bryophyllum. One more example is there. And that is of begonia. Now in begonia plant, the leaves are succulent, they are with buds, but there is a difference. This is the begonia leaf. In begonia, the buds are present on the surface of the leaf. See the difference? Margin of the leaf and surface of the leaf. Then you have to remember the example. The epiphylous buds, when they are present in the margin, then the example is bryophyllum, and when they are present on the surface, the example is begonia. The same condition is there. Leaves are succulent, they are with having stored food material, and adventitious buds or epiphylous buds which are present on the surface of leaf. During favorable condition, these buds will sprout to produce adventitious roots and the plantlets like this, and involved in the process what we call it as vegetative propagation. So these three are the methods of natural methods of vegetative propagation. Now, this part carries importance in your board exam because they used to ask one question for 4 marks, the long answer question. Discuss any 2 natural methods of vegetative propagation in flowering plant. There are 3. You have to write down only 2 for your board exams. As far as your NEET is concerned, many different level of questions, type 1, type 2 and type 3 questions. I will provide you the question bank. You will go through it and you can come across many questions can be asked on this concept. So this is the very important concept. You have to learn it carefully. Now the second type of natural method of vegetative propagation is vegetative propagation by means of stem. Basically, stem is the ascending aerial axis of the plant. But stem shows different kind of modifications. You have studied this part in morphology of flowering plant. Let us discuss. Stem is modified into 3 different forms, 3 different types. One is called as underground modification, then subaerial modification and aerial modification. Underground modification, in this case, basically stem is positively phototrophic and negatively geotrophic, but in some cases due to modification, due to adaptive ability of the plant, stem becomes underground, that is positively geotrophic, and such modified stems, they are related with storage as well as vegetative propagation, that is asexual reproduction. As we have discussed in the roots, they also show, such stems also show storage of ample amount of food material, and they are with the vegetative buds, or you can say axillary buds are there, because this is stem, presence of bud is the characteristic feature of the stem. So you are not going to use the term adventitious bud here. It is applicable to roots, it may applicable to leaves, but here the buds are either axillary or accessory kind of buds are present. So underground stem, they show presence of reserve food material, ample storage of reserve food material, and also they are involved in the process of vegetative propagation. The best example is the stem tuber of potato. See the diagrammatic representation. This is the potato tuber. It shows presence of eye. Many eyes are present on the potato tuber, This is the swollen part of underground branches. So definitely it is a stem and not a root. It shows presence of buds, and every eye, what we call it as these eyes, these are nothing but nodes. And nodes show presence of buds, or axillary buds are present. Now these buds, they get sprout in the favorable condition to produce the plantlet, like this. And in the natural process, the remaining part of the tuber, that will get degenerated, and these plants can grow as individual plant, individual organism. So it is the propagation by means of vegetative part, what we call it as stem, and therefore it is vegetative propagation by natural method, again by means of stem, and the stem is underground. There are many other examples like rhizome of ginger, bulb of onion, corm of amorphophallus. They are also show the same kind of vegetative propagation because the stems, they are modified underground structure. The second type, the second modification is subaerial kind of modification. Now subaerial, it indicates stem is not erect here, it is prostrate. It is creeping and crawling on the surface of ground, or you can say surface of water. See, this is the ground level. Now the plants which are growing here, having a creeping and crawling stem like this. The best example we are going to state here in case of subaerial modification of stem showing vegetative propagation, and that is Cyanodon, which is called as the lawn grass in Marathi, which is called as Durva. So this lawn grass, the lower branches, they are weak, creeping and crawling or prostrate in nature. They are just above the soil surface. So they are not completely aerial, neither they are underground. So they are called as subaerial modification of the stem. Importantly, these stems are weak and tender. And weak and tender nature itself indicate that these stems are not having the reserve food material, and hence subaerial modification of stem is only and only involved in vegetative propagation. There is no occurrence of storage of food material. Now, these weak stem, whenever due to their weak and tender nature, they get bent towards the soil. Whenever they get bent towards the soil and that touches to the soil, it will form the adventitious roots, and immediately the buds sprout to produce the new individual. Now when the connection is broken from the mother individual, the new plant can grow independently. So it is a propagation by means of stem, which is subaerial. Many other examples are also there, like offsets of Acornia, stolon of mentha or pudina. These are again the example of subaerial modification of stem showing vegetative propagation. So these are the two types, by means of underground stem, by means of subaerial stem. The last type is the aerial modification of stem. Aerial modification is meant for variety of purposes. It is involved in the storage, it is involved in vegetative propagation, it is involved in photosynthesis, it is involved in protection, and many more different kind of functions are performed by aerial modification. But our focus is there on vegetative propagation, so we are going to select the single example of aerial modification of the stem which is involved in the process of vegetative propagation, and that example is nothing but bulbils. Now these bulbils are the aerial parts, aerially developed structure of the stem. For example, let us see the example of Dioscorea. This is a creeper, climber, and having a weak stem. Now this climber aerially shows or forms some axillary structures like this, and these structures are swollen. It means they are having the reserve food in them. Again, as they are the part of stem, they show presence of buds. Now these are called as perinating structure. After death of this plant, those will get detached from the plant and in next favorable season they sprout as buds are present to produce the new individual. Bulbils are not present in Dioscorea only. It is also present in plant like ananas, the pineapple, and it is also present in cycas, which is one of the flowering plant belonging to gymnosperm. So these are the different methods of or natural methods of vegetative propagation by means of stem. Either they are underground, subaerial or aerial. Now this is time to discuss the artificial methods of vegetative propagation. Now as we have discussed in the previous part of the lecture, artificial methods of vegetative propagation, they are very essentially involved in the practices like horticulture and agriculture, or you can say in the field of gardening and farming, artificial methods of vegetative propagations are involved. Now these methods are classified into different types. One is called as cuttings, second is called as grafting, and the third one is called as micropropagation. Cuttings, as this is again a vegetative propagation, only vegetative parts of the plant they are involved in the process. Cuttings of vegetative part and using those cut pieces for propagation of the plant is simply called as cuttings. So these are the small pieces of either stem, roots and leaves involved for propagation or as a propagatory material. Cuttings you can done for stem, roots as well as leaves. Let us discuss. In case of stem cutting, now this is the mother plant with many branches. I want to produce the thousands of plantlets from the single plant. For example, a rose plant is there, rose bush is there, and you are going to produce a number of plantlets from the same plant. Then I will take these stem pieces, stem cutting of appropriate size. You can say the thickness, it should be there of your thumb size, because whenever you are going to use the stem pieces for a propagatory material, it must have the primary meristem. Presence of meristem is very important, and occurrence of meristem is seen in quite mature state, not in the young state, because that is not able to sprout, not able to develop. So appropriate size of the stem pieces are taken. They should be with primary meristem, and these pieces are taken for the propagation. Now see, when you select the single stem piece and you want to develop the plantlet and somehow it will lose, it will be killed, it will be get infected, many reasons are there, and there is a loss. Now this loss is quite affordable because you are dealing with a single plantlet, but when horticulture or agriculture is related, or cutting is performed for horticulture or agriculture, thousands of plantlets you are going to produce. So such loss is the loss of energy, loss of money, loss of time. So you have to ensure that all these stem cuttings, they will develop into the plants. For that purpose, these cuttings are treated with certain chemicals like auxins. The best auxin you can use is IAA. You can also use IBA. The names are there in your notes. Indole 3-acetic acid, indole 3-butyric acid. These are the naturally occurring auxins. Auxins are the growth promoting substances, and these are related with rhizogenesis. It means it is involved in the process of root formation, rhizogenesis. This term is not there in your notes. If it is not there, you have to write down there. It is involved in rhizogenesis. Now, what is the procedure? These are the stem cuttings. These stem cuttings are dipped into the solution of auxin, and for some time it varies from plant to plant, and these stem cuttings after treatment with auxin is then planted to the polybags having soil. Now each stem cutting will give rootings, no doubt, because they are treated with auxins, and these stem pieces will sprout into new individual, new plant, and the success rate is quite better. So stem cutting should be treated with auxins like IA and IBA, and this part is very important for your MCQ type of questions. It can be asked in your board exams as well as in your NEET exam. So this is the first type, which is called as cuttings. Cutting In case of stem is done for different plants like sugarcane, rose, grapes, etc. But you can also go for cuttings of roots and cutting of leaves. For example, sweet potato, peperomia. Peperomia is the leaf cutting example. Sweet potato is the root cutting example. Many examples are there included in your notes. This is the first part of artificial method of vegetative propagation, and that is nothing but cutting. You have to focus on auxins because this is very essential part. Presence of primary meristem in the cutting, that is nothing but presence of buds is very essential. So cuttings is the first part. Now the second part is called as grafting. The second method of artificial vegetative propagation is called as grafting. Grafting is defined as it is a process by which union of two plant parts is done so that they can grow further as a single plant. Now, grafting includes two different plants, and these plants are selected and certain systems of these plants are taken for the grafting. For example, the first partner in the grafting, you are going to use the root system of that plant, and the plant of which root system is used for during grafting is called as stalk. This is important. The second, you are going to use the shoot system of another partner plant, and that plant is called as scion. So grafting is nothing but union of two different plant parts, ideally what we call it as stalk and scion, so that they unite together and grow further as a single plant. So definition is important. This can be asked for 1 mark in your exam, in your board exam. These terms are very important for MCQ kind of questions. Now see how the procedure is that. So this is the rooted plant of which root system you are going to use. This is called as stalk, on which you are going to graft the scion. The stalk and scion, both the plant members, both the partners of the grafting, they should have or they should belong the closely related groups. Means they must have the common species. They may be variety of the same species, or they at least include to the same genus. You cannot graft a mango on the sapota because the biochemical constitution of these plant is different. So stalk and scion should be related with closely related groups, closely related. They should be closely related with each other. This is the first important thing. Second thing is stalk and scion, they will unite together and grow as a single plant, and that is only because of union of their cambium. So importantly, presence of cambium is the essential criterion for grafting, and therefore grafting is only possible in dicots because dicots show cambium, and in monocots you cannot perform the grafting. Now, this part is very important. This can be asked for MCQ or statement type of question, assertion reason type of question. Grafting is not possible in monocots. It is true statement. Why it is not possible? Because they lack cambium. This is very important point of the grafting. So grafting for which you are going to select stalk and scion, they are closely related groups. Then union of cambium is very essential, and therefore presence of cambium is must for grafting. It is possible in case of dicots. You cannot perform grafting in monocots. Now see the procedure. A scion and stalk, they are cut in a complementary way like this. Now this is the complementary cut made on stalk and complementary cut made on scion. Now this scion is the graft. It is with buds. Those buds will sprout after union of the plant parts. Now they are with cambium. Now this stalk and scion, they brought near each other appropriately like this, and grafting wax is applied over it, and it is sealed with the polythene tape. Now this is the site of grafting. Now one important thing you have to do is you have to remove all buds from the stalk. Because when buds are present on the stalks, see, these are the stalk buds. Now when these buds are present on the stalk, the nutrients are directly supplied to the stalk bud and your graft or your scion cannot grow. Instead of that, these stalk buds will sprout. So you have to perform this very importantly, that all the bud from the stalk should be removed so that only scion can grow and proliferate. This is the beauty of grafting. So simply, grafting is the union of two plant parts, and that union is mainly dependent on union of cambium. Cambium is the type of meristeme which is involved in formation of tissue in this wounded part or in this cut part, so that two plant parts will unite together and grow as a single plant. Importantly, plant should be of closely related groups, and presence of cambium is very must, and removal of all the buds from the stalk is the essential procedure in all type of graftings. So this is the grafting. In grafting, there are certain subtypes. For example, budding. Budding is nothing but grafting, is a type of grafting. So what is the difference between actual grafting and budding? In case of grafting, normal kind of grafting, you are using stem part, stem piece as a scion. But here in case of budding, here scion is not a stem piece, but it is only a bud. This is the important point. So budding is a grafting where scion is bud. This is the definition of budding simply. Now how budding is done? See, this is the stalk, which is the rooted plant. You are going to use the root system, so it is called as stalk. As a scion, you are going to use a bud. Now see, this is the plant on which this is a bud. I want to use this bud as a scion. So I will remove a patch like this. A patch is removed from the scion plant, and of same size, the patch is made here on the stalk. Now this patch of the scion bud is introduced in the cut piece like this. The rest of the procedure is similar to that of grafting. The grafting wax is sprayed and a polythene tape is wrapped over the grafting area. You have to take care that bud should be come out so that this bud will sprout to produce the new branch. So this is a grafting in which scion is a bud and therefore it is called as budding. The second subtype of grafting is quite interesting. And that is called as layering. In layering, there are two different types. One is called as ground layering and another is called as air layering. So these are the two types of layering. In ground layering, see the procedure. This is the mother plant in which the lower branches, see this is the lower branch. This lower branch is cut like this to expose the cambium. So this is the cut region, and this exposed region is applied or treated with auxins like IAA and is wrapped with cocoa pit, and this is then bent down and buried into the soil like this. Now the new plant which is developed here, after cutting this region, the new plant will grow as an individual plant. So this layering is called as ground layering. In Marathi it is called as dab kalam because it is bent down with pressure into the soil to produce the new plant. Now imagine this region is present here. In diagram it is not clearly shown. Now we assume that the treated portion is here, which is producing the root, and the new plantlet is developed. Now the second type of layering is called as air layering, as it is done in the air, or you can say on the aerial branches, not in the soil. For example, this is the branch, aerial branch of the plant. It is again girdled to expose the cambium like this, and treated with auxin, wrapped with cocoa pit and completely covered with polythene paper. Now this is called as booty kalam because booty is formed on the branch in Marathi. So this air layering, due to treatment of auxin, this cambium will show the rhizogenesis, root formation. When you cut this branch from the mother plant, you can grow this individual as the independent plantlet. So these are the types of layering. Is not included in your syllabus. Additionally, as questions are given there in your MCQ question bank, I have given that layering is of two different types, ground layering and air layering. See the beauty of the concept. Again here the treatment is done or is given by using auxins. So auxin treatment is very important in layering. So these are the different horticultural technique. Cutting, it is the very cheap and convenient method of artificial vegetative propagation, but grafting, budding and layering, it requires the skillful labor. The last method of artificial vegetative propagation is called as micropropagation. Now, earlier method like cutting, grafting, these are quite cheap and expensive. Some of them, they are required the skillful labor, but micropropagation, this technique is quite expensive, because it is the technique of plant tissue culture. The plant tissue culture strictly requires a controlled condition, laboratory condition, aseptic condition. So it requires so you can say it is the expensive method. The plant tissue culture technique, which is related with formation of large number of individuals from a very small part of the plant or small tissue piece, that is called as micropropagation. For micropropagation, you have to select the excised plant part, small piece of the plant part, and that is called as explant. Explant used for micropropagation, it must be shoot, apical, meristeme. See the plant system. This is the shoot, and this region is called as shoot apical meristeme. Now this shoot apical meristeme is separated from the plant, and you have to select it for formation of n number of individual by a technique what we call it as micropropagation. See the procedure. The artificial nutrient medium, culture medium is formed, is produced, and this culture medium is kept in a culture vial. This excised shoot apical meristeme, what we call it as explant, is transferred to the artificial culture medium, and it will allow to grow. The nutrients which are present in the culture medium are absorbed by the plant. Again, this is supplied with cytokinin. Now, cytokinin is the phytohormone. It is the plant growth promoter, which is having the role which is called as caulogenesis. Caulogenesis is nothing but it is involved in shoot formation. Due to presence of cytokinin in the nutrient medium, this shoot apical meristeme is able to produce number of shoot pieces, number of buds from a single shoot apical meristeme. Now, these pieces of the bud, they are separated from each other and shifted to the fresh culture medium for next growth. Now, this nutrient medium is supported with auxin, and you are already knowing that auxin is involved in the process of rhizogenesis. It is involved in root formation. So, these separated bud, or these are the separated shoot, multiple shoots, they form the root system like this. Now, these multiple shoots, already shooting is done there by application of cytokinin. Again, by treating with auxin, the root system is also developed In this way, from very small amount of tissue material, from very small amount of shoot apical meristeme, you can produce thousands of plantlet in a very short or stipulated time. This is the beauty of micropropagation. Only drawback is, or only limitation of this technique, it is expensive. It strictly requires the laboratory. Now, micropropagation is quite widely applicable nowadays in horticulture and agriculture because many commercially important plants like sugarcane, banana, grapes, etc., the plantlets which are available for the farmers in the market, those are produced by the tissue culture technique, which is called as micropropagation. Now, this is all about the artificial methods of vegetative propagation. Let us see the significance or the importance of vegetative propagation, or you can say artificial methods of vegetative propagation. The first significance of artificial vegetative propagation or vegetative propagation in general is it is related to the production of clones. So the plants are genetically identical, there is no change in their genetics, so you can use the plantlets for many generations. So formation of clone is the first thing. Second, you can recover the rare plants. For example, certain plants are facing the threat, facing the danger of extinction. You can collect those plants and by cutting, by grafting or by many other techniques like micropropagation, you can produce n number of plantlet to recover or to save that species from extinction. Then, next significance is you can propagate the commercially important species in a very short time. For example, plants which are not able to produce the seeds like banana and sugarcane, you can propagate those plants. You can produce thousands of plantlets from this very small material by using the artificial techniques of vegetative propagation. So important species can be propagated. Next one is propagation is season independent. For example, there is no need to wait for a favorable season or a particular season for propagation. You can propagate the plantlet in any season. So therefore it is called as season independent process. That is the important significance of the vegetative propagation. Next and very important significance is mostly related with grafting. Grafting is related with mixing of certain characters of stalk and sheath. Now focus on this point because we are going to discuss the vegetative propagation, which is the asexual mode of reproduction. And as we have discussed, in asexual reproduction or in the type of asexual reproduction, there is no mixing of the gametes, there is no variation, so you cannot see the variation, you cannot see the variants or hybrid in this process. Then how we can state that mixing of certain characters of stalk and sheath is possible in case of grafting? It is quite contradictory. Now let us discuss. See, this is the stalk of which root system you have used and on which you have grafted the sheath. The mixing of character is something related to the biochemical of the plants. Now see, stalk, suppose it is related with formation of certain biochemical compounds which is having importance in the disease resistance, insect resistance and many other characteristics. Now such biochemical, due to upward translocation of water, it is given to sheath also, and that is nothing but mixing of certain characters, or certain characters are shifted from stalk to sheath in case of grafting. These are the different significances of vegetative propagation. It is summarized there in your notes. That part can be asked for 2 marks if you are facing a board exam, and they can ask you state the significance of vegetative propagation. You can mention any 4 of the list which is given in your notes.